Today, we're diving into a really cool study that cracks open a huge mystery in brain science. Why do some of these devastating neurological diseases only show up as we get older? We're going to break down a major advance that could change how we understand not just one disease, but pretty much all age-related brain disorders.
So let's start with this question, because it really gets to the heart of it. For diseases like Huntington's, Alzheimer's, or Parkinson's, you can have the genetic risk your whole life, right? But the disease just sits there, dormant for decades. Well, this is the story of a team of scientists who finally figured out how to capture that one missing ingredient, time, and put it in a lab dish.
All right, so our main character in this story is Huntington's disease. It's this absolutely brutal inherited disorder. It's all caused by a single faulty gene. You might hear it called Huntingtin or HTT for short. And the weird thing is, the disease usually waits until a person is in their 30s or 40s before it really starts its relentless attack on the brain. And you can imagine this created a massive roadblock for researchers. I mean, they could take cells from a patient, grow them in the lab, hoping to study the disease and maybe test some drugs. But they kept hitting this wall. The very disease they wanted to study, it just wouldn't show up.
This slide really lays out the paradox perfectly. On the left, that's what's happening inside a patient's brain. You get these toxic lumps of protein, DNA gets damaged, and brain cells, specifically medium spiny neurons, start to die. But on the right, that's the lab dish. Using the standard methods, those same cells from the very same patient stayed stubbornly healthy. No clumps, no damage, nothing.
So, what was the deal? Why the huge disconnect? Well, it all came down to the method they were using. For a long time, the gold standard was creating these things called induced pluripotent stem cells, or iPSCs. Basically, you take a patient's skin cell and you use a special recipe to wind back its biological clock, turning it into this super powerful stem cell. It's an amazing technique, but it has one huge unintended side effect. And yeah, this was the problem. That factory reset completely erased the cell's age, basically wiping the slate clean. By making the cells biologically young again, scientists were accidentally getting rid of the most important ingredient, decades of life. Without that history, that wear and tear, the toxic proteins just wouldn't clump up and the neurons wouldn't die. It was a dead end. So this is where the big breakthrough happens. A team over at Washington University School of Medicine, led by Dr. Andrew Yu, they looked at this and said, okay, if resetting the clock is the problem, we need to find a way to keep it ticking. Their approach is so clever. It's called direct conversion. Instead of rewinding the cell all the way back to a stem cell, they use this specific cocktail of molecules to transform an adult skin cell from a Huntington's patient directly into a medium spiny neuron, the exact type of brain cell that the disease hits. And here is the absolute genius part. The new neuron kept the age of the patient it came from. This was it, the million-dollar question. For the first time ever, they had a neuron in a dish that was the same biological age as a neuron in a middle-aged patient's brain. So, would it finally get sick? Would it finally act the way it was supposed to? So, how'd the new model do? Well, as we're about to see, the results just cracked the whole case wide open. The answer was a total game changer. It was almost immediate. The researchers started seeing everything they had been missing for years. These age-retaining neurons began to show all the classic, tragic symptoms of Huntington's disease, right there, under the microscope. I mean, it was like they recreated the entire crime scene in a petri dish. All the key signs were there. They saw the toxic mutant huntingtin protein, or mHTT, forming those telltale clumps. They measured a ton of DNA damage. They watched as the mitochondria, the cell's little power plants, started to fail. And most importantly, the neurons just started to die, all on their own. And get this, the accuracy of their model was just incredible. The team found that about 10% of their lab-grown neurons got these toxic protein clumps. Now, 10% might sound low, but it's a near-perfect match for what's seen in brain tissue from actual Huntington's patients after they've passed away. This wasn't just some sick cell, this was a truly faithful model of the human disease. And I mean, look at this. It's a night and day difference. With the old time machine method, you get nothing, a clean slate. But with a new direct conversion method, every single sign of the disease just lights up. They had finally, after all these years, created a true disease in a dish. As the scientists themselves put it was that age-associated decline in protein homeostasis, that weakening cellular garbage disposal. That was the key. You see, the faulty Huntingtin protein was always there, in both cell types, but it only became toxic when the cell was old enough that it couldn't clean up its own mess anymore. And they proved it with this killer experiment. They took cells from the same patient and used both methods. The age-preserved cells, as you can see, developed those toxic clumps. But when they took those same cells and ran them through the time machine to make them young again, poof, the clumps vanished. That was the smoking gun. Age was the culprit. And what's really amazing is that the model didn't just show the disease. It showed how the disease progresses. The cells didn't just all die at once. Just like in a real patient, the degeneration happens slowly, over time in the dish. They had actually captured the tragic, progressive nature of Huntington's. You're looking at the rate of cell death over time. So on D30, both the healthy control neurons and the Huntington's neurons are doing okay. But look what happens. By day 35, you start to see them split. And by day 40, wow, the death rate for the age-retaining Huntington's neurons has just skyrocketed, while the healthy cells are totally fine. That's the disease, right there, unfolding in a dish in just 10 days. So, okay, the model worked. They could finally see the disease. But that just led to the next big question. Why? Why does an aging cell finally give in when a young one doesn't? The answer lies in this really fundamental process inside our cells called proteostasis. It's a bit of a mouthful, but think of it like this. Every cell in your body has a super sophisticated quality control system for its proteins. Its job is to manage everything, but most importantly, to clean up and get rid of any proteins that are damaged or toxic. It's the cell's essential cleanup crew. And this slide explains exactly why age is the key. In young cells, that cleanup crew is strong. It's efficient. It clears away the toxic Huntingtin protein before it can ever cause any trouble. But in the older, age-retaining cells, well, after decades of hard work, that crew is just worn out. It can't keep up with all the toxic junk anymore. And so that junk starts to pile up, poisoning the neuron from the inside out. So that leads to this huge realization. It's not like the faulty gene suddenly flips a switch in middle age. Nope. It's been pumping out that toxic protein all along. The thing that actually changes is that the cell's own defenses, its cleanup system, decline with age until they're finally overwhelmed. Time doesn't cause the disease, it enables it. So, what's the big picture here? What does this all mean for the future? Well, let's look at how this one discovery just opens up a completely new path forward, and not just for Huntington's. Well, the implications are just enormous. For the first time, scientists have a reliable way to test potential drugs on cells that actually get sick like they do in a person. They can finally dig into why these medium spiny neurons are so vulnerable when other brain cells seem fine. And maybe most excitingly, this whole age-retaining method could now be used to model other diseases where age is the main suspect. We're talking Alzheimer's, Parkinson's, and more. And you can see it right here in the words of the study's authors. They're basically saying this work proves how critical it is to include the factor of age when you're studying these diseases. By keeping the cell's clock ticking, we can finally create models that are true to life. And the power of this model goes even deeper. The researchers also took cells from patients who had the gene but didn't have symptoms yet. And just as you see here, those neurons formed the toxic clumps but had way less DNA damage and cell death. By comparing them to cells from patients who were already sick, they could basically watch the entire disease progression from beginning to end unfold in that dish. Ultimately, this changes everything. We've gone from looking at a single, static photograph of a disease to being able to watch the whole movie unfold, from the very earliest signs of trouble to the final, tragic stages. So yeah, this research solves a huge puzzle for Huntington's, but it leaves us with this incredibly hopeful question. Now that we have a tool to basically trap time in a bottle, what other devastating age-related diseases are about to have their secrets exposed? What else is running out of time?